Perfect. Is this on?
Okay, he'll tell us about spatial simulation analysis of actin filament dynamics and wave propagation.
All right, thank you. You can hear me okay? Okay, not screaming or anything like that.
Turn the switch on if it's not red lit.
Is it on?
It's on.
Are we sure?
Yep.
Okay, good. Okay, so again, my name is Sanft. A lot of you don't know me, so let me just spend one slide introducing myself, but also help you give you some of a perspective of where I'm coming from. So I graduated from Linda Petzold's group. I'm a computer scientist, and I worked with Dan Gillespie along with Linda, and did things like multi-scale methods for discrete stochastic, but I see a lot of things through a Gillespie lens. And at the time, I was doing a lot of non-spatial stuff. After that, I went to a place called St. Olaf College in Minnesota. So on the map here, Minnesota is up here in the northern United States. That's where I'm from originally, so for family reasons, I made my way back there. So St. Olaf College is a small liberal arts college, which means it was a lot of teaching. And while I was there, I met a nice applied mathematician that I developed a relationship with. And so then after my visiting position was done, I wanted to stick around. Her name was Becky Vandiver. She was actually a student of Alain Gorielli, who's now at Oxford, so not too far from here. I wanted to stick around, and so I ended up going to the University of Minnesota, where I worked with Hans Othmer, who I think spends a lot of time here. A lot of you probably know him. And then Becky and I, in the last year, we got married and solved the academic two-body problem with constraints, which we're pretty proud of. So we're now at the University of North Carolina at Asheville. So a lot of you know University of North Carolina. Actually, it's a part of a system of schools. And University of North Carolina Asheville is the designated liberal arts university in this UNC system. So North Carolina is down here in the southeast part of the United States, and Asheville is in the western part of the state in the Appalachian Mountains. Beautiful town. We're super happy there. But because it's a liberal arts university, that means I have a heavy teaching load, which means I don't have as much time to work on research as I'd probably like. It's purely undergraduate, so I don't have any graduate students, which means I have to do a lot of collaboration to get people to do work for me. I mean, get work done. Okay, so the motivation, let me get to my talk now. This project is really motivated by cell motility. So this is a collaboration with Hans, sort of extending some of the stuff that we were doing when I was a postdoc with him. He's very interested in cell motility just in general. So from things like how do cells divide to things like cancer metastasis. And so we're looking at actin. And I'll talk a little bit about actin and give a little bit of background before I describe our model that we're working on. And then being a computer scientist, I'm really interested in the simulation challenges that this model presents itself. So I'll talk about that. I'll present some results, but what did you call it? A sheepdog story? This might be a little, I thought, what was it, sheepdog? Shaggy dog story. The results are still very preliminary, and so take it for what it's worth. Okay, so cell motility. Here's a famous video from 60 years ago or something of a neutrophil chasing a bacterium. I don't know if this is the same microscope that you've been using. Pretty much the same. But what you see, right, is that this neutrophil is chasing it down. So you see how in the front there's something happening. It's directing its motion towards some signal that this bacteria is emitting, and eventually it chases it down in this movie. And so for me, being a computer scientist, this kind of thing is fascinating, right? Because this is like chemical, right? It's not like I can envision a mammal, like a wolf chasing its prey or something and sort of thinking about it. This is all chemical. And so the system that we've been using as sort of our model system is DICTY. And this is interesting all in itself as well. It has an interesting life cycle. So maybe I'll start up here. So in one stage of its life cycle, it's the single-celled amoeba. And a bunch of them, they cruise around and they feed and they go about their business. In the absence of resources, so if they start to reach a starvation state, a signal will they'll send out a signal, and this big group of these amoebas will cluster together, aggregate into this lump, and this lump will actually turn into, I guess for lack of a better word, a slug that'll crawl away. And then that slug will grow a stalk and this fruiting body that releases these spores to explore new territory. So it's really fascinating for a lot of different reasons. And here's a little video of these amoebas, and you see for a while, the resources are plentiful, and then at some point they'll hit a starvation state, and you can't really see the signal, but you'll see the result. And the result is that when they sense this signal, they start to move toward it, and you get this aggregation. Very interesting. It'll become obvious. Like right in here, something will happen, and you'll start to see everything biased toward that direction. So now you start to see it. Right? So you have all these individual amoebas who were on their own and happy, now are coming together to form this aggregated slime mold thing. And eventually this thing just crawls away. It's a little bit gross, but fascinating. So there it goes. So this is an interesting system for a lot of different reasons, including there's sort of some self-sacrifice that goes on. Like some of the individuals sacrifice themselves for the betterment of the colony, and at some point I think it kind of goes out of frame, but I think it does grow this stock. Yeah, so this dicty is very interesting for a lot of reasons. We are mostly just interested in single cell motion, so we're not so interested in this aggregation or this slug moving around. So single cell motion is complicated, and there's a lot of things going on. One important part of that is actin polymerization in the direction of motion. So actin is this monomer that forms these polymers, and the intuition is that these polymers sort of grow in this direction that the thing needs to move and kind of pushes it in that direction. And then you need also some things in the back to contract and bring the back forward. And there's been a lot of work on actin. Several groups have mentioned it already. And in the literature, several groups have published work on actin waves, including people sitting in the front row. Hans looks up to Gunther Gerish's work quite a bit. I think he's pretty famous in the field. He's shown, among other things, waves in PIP3 and PTEN and their correlations and anti-correlation with actin waves. But the basic quote that I want to focus on is these actin waves propagate along the planar surface of substrate-attached cells. And so there's a lot of different things that we could be looking at getting toward single-cell motion. We are just focused on some actin dynamics. So it's really a very simple model that I'm going to be describing. Okay, so some background. So actin is a protein that can be available in monomeric form, known as G-actin for globular actin. So that's an actin monomer. But these monomers can polymerize to form filaments. So for some terminology, I'll often use monomer for a single G-actin monomer, and then polymers or filaments or backbones or branches to refer to these polymerized strands of actin. And so here's a little picture of an actin filament. In some of the cartoons that I'm going to show, in my mind, I visualize this polymerization as, okay, an actin monomer is a circle or a sphere, and when they polymerize, they just stack on top of each other. That's not totally true. That's the cartoon picture. So you can see there's sort of a little bit of a rotation and a staggering. One important thing to mention is that there's two different ends, the barbed end and the pointed end. And generally speaking, polymerization happens at the barbed end, depolymerization happens at the pointed end. So there's these two ends, different things are happening at the two different ends. It might be. I'm not going to count. So in addition to forming these filaments, they also form branches. And that's mediated by ARP23. And so you can see in this picture right here, we have one filament here and another filament branching off at a fairly specific angle. Here's like the mirror image of that. The idea is that there's these things, nucleation promoting factors that combine with ARP23 and actin monomers. These things combine and form these branches, right? And the NPF sort of changes its state. So I'll be talking about NPF, I'll be talking about ARP23 a little bit, but the important thing is that we get these filaments by polymerization and we get branching. Okay. And to give you some insight on how this process produces some force, there's this little cartoon here where you see the membrane, again, in a lot of cartoons, it'll be sort of, looks like it's rigid and fixed, but there's some wiggle, and as these filaments grow, there's a force applied. just to give you some sort of insight. We actually are not looking at any of the forces applied in our simple model. So let me get into the model. Doing a stochastic model, so like a Gillespie type of thing, in a 3D Cartesian domain. So the typical dimensions are about 5 microns by 5 microns by 2 microns divided into a grid of roughly 50 by 50 by 20. And the way I visualize it is the bottom of this box is the membrane, and that membrane is affixed to a substrate. Scattered along this membrane are various nucleation sites where backbone filaments can grow. And so in the model we have 9 species, so independent species, and then what I'm going to call local actin clusters. Local actin clusters are these actin filaments and branches. So the species we have in the cytosol G-actin monomers, so the actin monomers are diffusing around in 3 dimensions. ARP23 is as well as coronin, which mediates some of the effects, has some influence on the shape of these filaments, and a capping protein that binds to an end. The nucleation promoting factors exist on the membrane. They can diffuse, and they're around in 3 states. So NPF, it's normal base state, an activated state, and then a recovering state. Where the activated state is how these branches form. NPF can bind to ARP23, and that complex can bind to G-actin, so kind of like that cartoon that I showed before. Now, these actin clusters, that's where the real interesting part of the model is. And so let me just try to give some intuition on what we're doing without too much detail. So the filament reactions. We have a backbone filament can nucleate on the membrane at one of these nucleation sites. And then you get polymerization at the barbed end, so that's actually the side on the membrane, and depolymerization at the pointed end, which is away from the membrane. So just as a simple cartoon here, if you think of this line here as the membrane, here is say a nucleation site, then these little actin monomers are diffusing around. Oops, oh, my movie's not going to play? Oh, it is playing. On my screen it's not. Okay. Okay, so what you saw is these things are diffusing around. Eventually, a monomer nucleated at this site, and then when another one bound to it, it sort of shifted it up. So again, my cartoon makes it look like these things are stacking on top of each other, but there's actually some offset, and also there's a difference between a monomer, a dimer, and a trimer at this bottom scale. Okay, so these things move around. These are all diffusing now. Again, the polymerization is happening at the bottom, effectively pushing this thing up. And you get the point. Now, at some point, we have depolymerization happening at the top, and this is sort of the basic process of how these backbone filaments get started. Okay, so just sort of a cartoon picture. There are ways in which this cartoon picture is different from our model, and there are ways in which the model is different from reality. For one thing, we think of these things as sort of rigid structures when they're not. So here, this little shape here, think of this as our nucleation promoting factor. And the nucleation promoting factor can move around. So it's on the membrane, so it can move around in 2 dimensions. I'm just showing a one-dimensional view. Now, at some point, if this nucleation promoting factor becomes activated, now the ARP23 and the actin monomer are able to bind to this nucleation promoting factor. And so yeah, it's moving around in 2 dimensions on the membrane, and at some point you get this binding of both ARP23, which is not explicitly showed here, and this actin monomer, and then that enables this thing to form one of these branches. Right? So one of these ARP23 branches like you saw before. So the membrane is down here, so this branch should be at an angle kind of like this. In our model, we don't take into account the branching angle. We sort of, in a very simple way, think of these things as sort of growing vertically straight up from the membrane, which is not entirely accurate. And then after this new branch forms, the nucleation promoting factor becomes inactive, or recovering. So there's a period of time in which it's not able to be reactivated. So there's this sort of resting period, I guess.
So the branching is only taking place at the membrane?
Near the membrane. So we have a small activation zone where that can happen, yes. Okay, so those are the basic filament reactions. We have an activated NPF, an ARP23, and a G-actin combined. I've written this as sort of one step, but this is actually two reversible steps. But that complex of these three things forms this new branch, and recovering NPF is sort of spit out of the process. One interesting thing about our model is that we include a sort of positive feedback term where free barbed ends, so these are barbed ends that aren't capped, can help activate NPF. So you get activated NPF creates new branches with new barbed ends, and those barbed ends activate more NPF. So there's a little bit of a positive feedback thing happening. And from my perspective, the really interesting thing about this model is that you have branches that are connected to branches. If you have a barbed end up against the membrane that polymerizes, it sort of pushes that whole network of branches up. So if you can visualize that. And then, like I mentioned before, we're not considering the effect of this angle, at least not yet. Okay, so let's talk about how we simulate this. So it doesn't quite fit into the standard Gillespie algorithm framework. So in the Gillespie algorithm, if you think of the non-spatial case, we have some number of species, some number of reaction channels, right? And if we extend it to the spatial case, like the RDME case, we have some number of subvolumes, right? Where algorithmically solving the spatial case is algorithmically essentially the same as the non-spatial case, just a relabeling of what you call species and what you call channels, and adding some more channels for diffusion to adjacent subvolumes, right? In our case, so species, what we're thinking of as species, include things like the barbed end and a pointed end of one of these filaments. So that's not necessarily a problem. But what is a problem, I think, is that you can have a reaction, so an event, in subvolume X that can push a species that's currently in a different subvolume Y into subvolume Z. So this would be the example if you have some long filament, you have a polymerization at the bottom, pushes everything up. Right? So in the standard Gillespie algorithm, or the spatial version of it, everything's local. Right? You have a reaction happens inside a subvolume. In the worst case, you have diffusion, which affects the populations of two adjacent subvolumes. So this is a different situation. You need a little bit more information than the typical Gillespie setup. So, one way to look at it is you can think of the channels. So I'll say channels as a generic term for both reaction events and diffusion events. So I'm not thinking of like calcium channels or something. I'm thinking of the types of events that can occur. Those types of events that may occur and their rates might depend on various attributes of these filaments. And in some settings, you see people who try to simulate these things by treating, say, a filament of length 3 differently as a different species from a filament of length 4, and so forth. That's fine if you want to sort of show like a toy, really large problem, but it's not necessarily the best way to treat that. And the other thing that I think is most important is the state change vector. So typically in the Gillespie algorithm, when you fire a reaction, it updates the state in a specific way. Right? And that tells you what rates you need to update, for example. In this case, the state change depends on the branch connectivity. Right? So if I have a polymerization, let's say, that pushes this thing up a little bit, that might change this. If I have several branches, that might change several things all in one step. And it's going to depend on the connectivity of these branches. So in a way, it kind of reminds me of this old moleculizer software, if anybody remembers that, where you sort of create species on the fly. But more so, I think it reminds me of rule-based modeling. Right? That seems like the way to do it. Like there's some rule based on the current state that tells you how you update your system. But important for us is that we still assume that each of these reaction channels behaves like an exponential random variable. Right? Like that's the key to the Gillespie algorithm. Right? You make this assumption, and then the Gillespie algorithm follows. So we still make that assumption. And so if you are doing a non-spatial model, a small model, you probably start with Gillespie's direct method. So I'm assuming most people here are familiar with the Gillespie algorithm, but I just want to sort of talk about it to put it in perspective a little bit. In the direct method, you choose the step size as an exponential random variable with a rate equal to the propensity sum. So by propensity, I just mean a rate. So each reaction channel has a rate at which it's firing. Gillespie calls them propensities, so I call them propensities. Yeah, and this is just a property of exponential random variables, right? If you have m exponential random variables, the time to the first is also an exponential random variable. And now to choose the reaction index, the typical way that this is implemented is you choose a uniform random number between 0 and the propensity sum, and then you iterate until your cumulative propensity exceeds that random number. Right? So the intuition for me, at least, is suppose you have these propensities, which I'll label A1 through AM, stored in a one-dimensional data structure, like an array in C. When you choose this random number here in that range, essentially what you're doing is you are pointing toward one of these reaction channels. You just don't know which one it is. Right? And so then this loop that iterates until the cumulative propensity sum is greater than that random variable is just figuring out where that is. Right? So I start at the beginning. Is the sum I've seen so far greater than that number? No. Then I move on. Right? And at some point, that cumulative sum exceeds a random number, and that tells you, oop, that's the reaction that happened. And then you go from there. But there's no reason why you have to store these things in an array. Right? You can do different data structures. In particular, you can group them in arbitrary ways. You just have to keep the sum of each group. So for example, let's say I have 8 reaction channels. Instead of storing them in a one-dimensional array of length 8, I can store it in 2 arrays of length 4. So whereas in my length n array or length m array, I have to go roughly halfway if these things aren't ordered in order to find the next reaction event. If I split those into 2 groups, then I cut my average search depth in half, basically. I first have to search to figure out which of these 2 groups it is, but once I do that, I can do a shorter search. Does this make sense? It sort of intuitively makes sense. And these 3 pictures are just 3 ways of viewing the same thing. So you might implement it like this, where you have a sum of everything beneath it here. If your random number exceeds that value, you go here. If it's less than that value, you go down here and do your search, your linear search that way. You might store it as a tree. I visualize it just like this, knowing that I need to keep those sums somewhere, right? But we can partition these things in arbitrary ways. So if we take our 2 groups and take each of those groups and partition them into 2 groups and keep doing that, we get a binary tree, right? And then the search and the update of our data structure is logarithmic. We could take what theoretically would be the optimal 2-dimensional orientation, right? And group it into the square root of m groups of size square root of m. Or you can do this, and you can keep doing this arbitrarily, right? And you can define all these different methods for statistically equivalent to the original direct method. You can also partition these groups by powers of 2, and then sample the group exactly like I was doing before, and then use a rejection algorithm to choose the particular event in the group. You do that with some assumptions on how these things are distributed. You get a order 1 constant complexity algorithm. So it doesn't matter how many reaction events you have, your rejection scheme basically lets you have the same amount of computational effort per step. So that's beneficial if you have large enough problems. And then there's also next reaction method variance. So this is the idea of instead of choosing the time and then which event separately, you generate the event time for all of the reaction channels and put them in a priority queue. Right? So typically, you think of implementing a priority queue with a binary heap. Right? So you have a binary tree structure where the smallest value is always at the top. That ensures that finding the next event is constant complexity operation, right? It's always at the top. And updating it is a logarithmic cost event to maintain that heap structure. But you don't have to use a binary heap. You could use a one-dimensional array. So a priority queue is an abstract data type, right? It has an insert method and a extract minimum method, right? And how you implement that is it's dependent on your implementation. So you can implement it with an array, right? I can insert it into the end of the array, and to pull out the smallest, I just search over all the elements and see which one is the smallest, right? Then I get linear complexity in the number of events. Or I can use the binary tree, and I get logarithmic. Or if you see our paper with Hans from last year, you can bin your events into time ranges and effectively get a constant complexity next reaction method. The next subvolume method is probably the most widely used method for this type of simulation, and it's very effective. Basically, it groups events by subvolume, right, using the next reaction method idea, right? So you select from which subvolume is the next reaction going to happen? Once you've chosen that, then you effectively do a two-dimensional search. You choose either a diffusion event or a reaction event, and then which one of those are you going to do, right? So if nothing else, this paper here, I try to put all these different methods sort of in perspective as just different ways of grouping the reaction channels in a way that's beneficial for a particular problem. Right? So that's foreshadowing what we do. So we basically do the opposite order of the next reaction method to simulate this system. So we first group by reaction channel, and in our model we have 25 reaction channels, including diffusion, and we select the reaction channel first, right? So we might choose, the reaction that we're going to fire next is a polymerization event, and then we go find where it happens. And to find where it happens, we do a two-dimensional search on the membrane, right? So we keep sums of the membrane, all the events that could happen at the membrane and above it, and actually we group these things so that our search is optimal, right? We can vary these parameters to find the minimum, so that's what we do. In the z direction, if it's not a membrane reaction, because some of our reactions are stuck to or restricted to the membrane, then we search up the z dimension until we find it. And this is particularly useful because we might say maybe it's a depolymerization of some branch, right? Well, now we have to search and find which branch that is. We start in a particular filament, right? And then we can search the sort of attributes of that filament to find where that event happens. And the beauty of that, and the reason this is beneficial, and the reason we do it, is because then you know what filament you're on, right? And so all of our updates are on that filament, right? If something's changing in this filament, we need to know what that is and know everything that's connected to it. So we end up with something that kind of resembles object-oriented programming. So I'm a computer scientist. Object-oriented computing is where you take your data and you take the operations that you can do on your data and you group those things together. So that's kind of what's happening here. The objects are these local actin clusters with filaments and branches, and the operations are things like polymerize, depolymerize, or cap, or unbind the ARP23 junction, or things like this. So this is what we do. Let me just talk about what our model shows. So without any activated NPF star, we just get some backbone filaments that amounts to just be noise, right? So we don't get any sort of spontaneous interesting happening. We don't have a mechanism for that. But if we start with some activated NPF star, which we typically do in the bottom left corner, then we get these waves of actin that propagate across the membrane. So let me just show you a little animation. Right, so we started by activating some NPF down here, and you see that this wave propagates to the domain. And again, the membrane is like attached to a substrate or against a substrate, and when it gets to the edge of that domain, the waves just die, which is what some experimental groups have found. So let me just maybe show that again. So what are we looking at here? So the way to visualize this is that we're kind of looking down on the membrane. And so the intensity there is some measure of the amount of these actin filaments, right? And so you see the actin filaments are sort of highest along a front, and then they sort of die off behind that wave front. Right? Okay. So what we'd like to do is take a simulation like this and try to quantify what we're seeing in these waves. It's the wave dynamics that we were really looking for based on some other published work. The wave speed, pretty easy to measure. And as you might expect, if you followed some of the things that I was saying, it depends a lot on this NPF star diffusion rate. It's actually the diffusion of activated NPF that's making our wave move. If we incorporate this branching, that might add some additional forward speed to our model. But we don't have that right now. And the amount of free NPF depends on a lot of factors. It depends on how quickly it's being absorbed and how long the recovery rate is and things like that. But the wave speed itself is easy to measure. The wave height, and in particular the wave shape, is a little bit more difficult to assess, and especially when we're trying to compare against experimental results. So the reason we made this video the way we did is we're kind of trying to mimic what you see in turf imaging. In the turf imaging, you get sort of a small slice near the membrane but not necessarily great resolution of what's actually happening inside that slice. One important thing, the number of adjacent actin clusters strongly influences the shape of a particular cluster. So if you just had, say, a single actin backbone and branches, it would tend to grow much higher than if it is near a whole bunch of other ones, because you get these other branches sort of absorbing a lot of these actin monomers. So you get different behavior in this sort of spatial setting than you would if you tried to study the local dynamics in isolation. So Hans and I talk about this a lot. He's like, what are the local dynamics? I'm like, well, this is what it would be if you make some assumption, like if I assume a constant amount of G-actin monomer in the cytosol, but that's not the case if it's part of this larger sort of interconnected network. So you can't study the local dynamics in isolation. So if we go back, so this is just a snapshot from that movie I showed you before. I just want to look at, just arbitrarily, this first column. Not because it's anything special, but just because I want to look at just a slice of this data. So what are we showing here? So one thing we could show, right? This is a simulation, so we have all the data that we need. Right? So we could show, for example, the total number of bound F-actin, or say the total number of monomers that are bound to filaments. Right? That's one measure of the shape of this wave as it's moving across. And what you see here, and this is not necessarily, the fact that this thing has these spikes is not necessarily representative of the typical case, but this is what it is for this particular snapshot. That's one way in which you could characterize the shape of this wave. But as you think of, what I said is our pictures, we're looking down on the membrane. Maybe a better picture would be how high are these things reaching, right? So I said these things are growing up. How high are these reaching? That's a different thing than the number of monomers that are bound, because it depends on the branching, right? If you have 300 monomers next to each other, like two branches with 300 monomers next to each other, you're going to have a height of 600, right? And so we don't get all the information here. So if we think of just sort of looking down from above, what's the maximum height, we get a slightly different shape. And so remember at the front, so this would be the front of the wave, and this would be the back of the wave, where the subvolume here corresponds to that bottom left corner and the bottom right corner. What this is saying, remember this front was very high before, but the actual height is lower here. That's suggesting that it's more dense here, but doesn't reach as high. So sort of interesting. You want to view sort of by position. You can do, this is a jitter plot. It's not exactly representative of what our model shows, but it gives you the right idea that, yeah, up in this region near the front of the wave, you get some dense pockets, and things are less dense, and you can even have these little holes. That's we'll call it a feature of our model. And these lines that I showed here are the subvolume, the vertical subvolume divisions. So this is not to scale. So the subvolumes in this direction, there's 50 of them, and here you only see, 7 or something like that. They're actually the same size. So this plot is not quite to scale, but it gives you some idea of the distribution of bound actin monomers. Okay. Okay, but I said we are interested in trying to recreate turf imaging, and turf imaging just takes like a small slice near the membrane. So what I want to do is just sort of look at everything below here. So basically everything in that first vertical subvolume. And if you do that, you get a plot that looks something like this. But now this is only showing the, let me go one more. This is only showing the bound monomers, right? If you're trying to image this thing, you don't only get to see the bound monomers, you get to see sort of all the monomers. And so if you add that noise that you would see from these free monomers sort of floating around in the cytosol, this is the signal that you would get. And so that's essentially what we're plotting in these movies. And what we like to do is match this up with both turf imaging, which gives us some information, but also other things that are known about these actin clusters. So let me just show maybe a couple other animations here. So this is just another simulation run. What you see is this little actin spot forming behind the wave, and that's something that they see in the experimental studies. Sometimes they die out like that. Other times they propagate and form these messy waves that move around. We've tried other things. Like you saw, the wave basically dies when it hits the edge of the membrane. Here we have activation in all four corners, and you see the wave also kills itself when it runs into itself. So this is another thing that they've seen experimentally that we're just trying to get a handle on understanding why this is the case. And Hans is interested in other things like can you get spiral waves and things like that just for the fun of it. And so we've kind of tried that a little bit by changing the initial condition, but so far we just get some messy stuff. So I don't have anything interesting to report as far as spiral waves go. But let me just basically stop there because that's where we are essentially. But let me talk about where I think we're going. In the short term, we're definitely working on refining this model. So there's things that a lot of things that you could add, a lot of things that we currently have that you might want to tweak a little bit, and we like to do things like sensitivity analysis and see, well, to what extent does it matter? And we really like I said, match the wave shape, including the density of these actin filaments. We also have done some ODE analysis. So basically what you get, if you convert this to a deterministic model, you get an excitable system. But for certain parameter ranges, it looks like you can get some oscillations, and we're trying to understand the regime in which those oscillations arise, and if you can get those same sort of oscillations in the stochastic setting. And then my personal interest is I'm really interested in developing tools to simulate models like this, because like I said, it's different from the standard Gillespie setup. And the idea with what I'm working on is essentially trying to do discrete stochastic, but with objects. So you define some interface for the spatial solver that interacts with some objects, like in C++ or something, and also geometry, and these things talk to each other in a reasonable way that'll allow you to basically swap out the solver. So right now we have this hard code, like homegrown solver that we use. We select the reaction first and then find out where it happens. We'd like to be able to just take our model and plug it into different solvers for efficiency reasons, really. So long term, another advantage of having these tools for simulation is that then we could change our model. So right now, one of the reasons that our branches grow straight up is because it's difficult to keep track of the position if you have sort of arbitrary branch angles and length. So utilizing our simulation advances to have more accurate branch dynamics. And then where I think we would like to go is right now we're not talking about any sort of force. This wave is not generating any force. But I said this project was motivated by cell motility, and so what we would like to do is ideally match up some biochemical modeling with some mechanical modeling to understand how these actin polymers push this thing forward. And for me, perhaps some new collaborations. So I teach a lot during the academic year, but summer is great. And because I'm at a teaching-oriented institution, I think there's maybe some interesting opportunities for pedagogy and undergraduate involvement, so sort of the broader impacts kind of thing. I'm also on leave in the fall, which is great, but before I get too excited about having all this time to do research, the reason I'm on leave is to take care of this little guy. So it turns out he takes a lot of time. But so I should acknowledge, so this is work with Hans. Some of it while I was a postdoc and in his group was supported by his NIH grant. Yugan Chen is also a collaborator, although he just took a job for working in industry at a pharmaceutical company, so that's kind of slowed down the progress on this project a little more than what we would have hoped. So that's it. So thank you.
Questions?
Yeah.
So you're treating each filament as an individual identity? Or
Yes. And you can think of it as each filament and its attached branches as an individual entity, effectively. So basically like a linked data structure that...
Have you thought about having a model where you do a more GSP style thing where you just remember the counts of, I don't know, branches or the average length? So you don't have to,
No. Because at least right now we're really interested in how high these things get, and we need information. So we have to first do it basically what we're doing before we can start doing things like that, I think. Yeah.
And maybe you mentioned is how fast is the kind of simulations you showed, like
The time scale?
like in terms of computation time?
Computation time? Basically, it takes a long time because there's a lot of actin. So we're doing full discrete stochastic simulation. So basically, I just run them overnight. It takes a long time.
Can I ask a question?
Sure.
So you played with the PD, like the mean field, and if it's excitable, it predicts the velocities of like square root d over diffusion coefficient over the kinetic rates, and then you check the velocity of the stochastic simulation. Do they match?
They do not match, but I don't remember in which direction. So no. That's as much as I have off the top of my head. Although I can get you more information if.
But anyway, you're saying stochastic effects determine wave velocity, not determine.
They influence, yeah, they influence it. I think so. Yes.
A question about, I think you're including the ARC-2-3 for the branching, right?
Yes.
And so does that, and to confront it with in vitro studies, you don't have to worry about like motors and everything like that because they're not including them. But does the ARC-2-3 also connect separate filaments substantially, like at the concentrations you're using?
Sorry, say that again.
So does it connect these? I mean, you have computationally distinct elements, and they're not growing under any stress. But if you start gluing them together and they're starting to grow independently, you're going to start having just stress effects.
Yeah, so we don't
interaction.
include any of that mechanical.
But the question is should you be including cross-linking between individual filaments at the concentrations of ARC-2-3 that you have?
We do to some extent. I mean, if you have, say, two branches that are attached to each other via an ARC-2-3 junction, right? So it should look kind of like this. We draw it kind of like this,
Sure.
and the polymerization down here pushes them both up.
Right.
Does that answer your question?
But you're not cross-linking them after they're away from the basal membrane.
No.
And my recollection was that ARC-2-3 can do that. Is that not kinetically relevant?
I'm not sure.
Okay.
Not sure.
Other questions? Okay, so before we thank our speaker again, I'll tell you the timeline. We're going to take a break now, and then at 4 we'll come back for the Rothschild lecture, and I'll ring the bell. Okay, thanks, both speakers.
Thanks.